Hello, my name is Nikhil Krishna, and this is the VR view of a nucleosome of PDB-ID 1A01. So we are going to have a look through the structure of the nucleosome, the properties of it, and some unique insights that I have observed uh, through VR into this nucleosome. So first of all, nucleosome is a part of the nucleus in which DNA is tightly coiled around. As you can see, this is the pink color chain is the double-stranded DNA to which it is wrapped around a huge protein complex. This complex is known as the histone 8 complex since it contains 8 different chains. So I think it would be better to show you if I colored each chain by its color. So as you can see, there are 8 different histone chains, and DNA tightly binds to it and creates one revolution around the histone protein, and this pattern basically at the edge right here. This pattern again repeats from here on. Hence, millions of nucleosomes are packed into one chromatin of DNA. So I'll add coloring to the heterocyclic atoms of the complex, and here as you can see all the atoms colored according to the chain color is carbon atom, and red color ones are oxygen and blue color ones are nitrogen, and if we by rare see any yellow color atoms, those are sulfur, and similarly phosphorus is usually colored purple, and iron is generally colored brown. So in this case, we actually have a DNA which is wound around this protein. So how does the DNA actually get attracted to the protein complex and stay in this position? So we are going to have a look at the electrostatics of nature of the residue and of the DNA nucleotides as well. And we are going to find out if any non-bonding interactions such as electrostatics or van der Waals interactions, London forces play a major role in this interaction right here. Since as you may view that there is none of covalent bonds present between the DNA and the protein. So if I just draw the surface of the electrostatic first by selecting the nucleotides and then drawing the electrostatic surface for the DNA. This may take some time. As it's present here, you can just see the whole coil is colored red, since in this coloring scheme, blue represents a positive electric charge, and red negative represents a negative partial charge. And this negative charge of the entire DNA originates from the phosphodiester bond, which forms the backbone of the DNA. If I just remove the surface right here, and let me hide the cartoon. As you can see, DNA is formed of these repeating units, which is a phosphodiester bond between each nucleotide, which is bound to a sugar, usually ribose in this case, and a purine or a pyrimidine. In this case, this is an adenine, and this is bound to a single cyclic thymine right here. So this is a sugar molecule. In this case, in our DNA, it is ribose, and this is a cytosine. Sorry, this is a guanine, double heterocyclic guanine, which is bound by a double hydrogen bond to a thymine. So this blue dash connection right here represents a hydrogen bond which attracts a partial charge right here at the nitrogen of the guanine, which is connected to a hydrogen. I think I have not shown you guys the hydrogen right. So to actually view the hydrogen bonding along with the complementary partially positively charged, sorry, negatively charged atom. We will first of all add all the hydrogen atoms within these nucleotides by going to, so we have to go to the tools tab, to the structure editing tab, in which as you can see there's a option called doc prep, in which we can prepare the entire complex, in this case for docking, which basically adds hydrogen, deletes solvent, deletes other non-essential ions, and fixes residues, and importantly adds partial charges to the whole complex, so we can view electrostatic interactions much more easily. First of all, let me just deselect the entire complex, and we will just prepare doc prep with the default model, and adding hydrogens takes a little while. We'll also assign charges using the Gastinger method, which Chimerax uses by default, and we do not need to save our molecule right now. So now it's pretty clear that this nitrogen within the guanine has two hydrogens bonding to it, and the partial negative charge of the nitrogen, if I can select the atom and use electrostatics, as you can see, this nitrogen forms a very strong negative shell around itself, because nitrogen is an electronegative atom with uh, 5 electrons in its outer shell, and hence it needs 8 electrons to become stable, and this additional 3 electrons it shares along with the hydrogen it's bonded to, and the core atom as well, the core carbon atom right here within the heterocyclic ring. So this negative charge actually is part of the nitrogen, which attracts electrons from the hydrogen towards the nitrogen. This creates a partial positive charge within the hydrogen, and already within the cytosine there exists a sole oxygen atom which is bonded by a double bond right here. And again, since oxygen is a even more electronegative atom than nitrogen, it attracts electrons from the core carbon atom to the oxygen, which then forms this hydrogen bond, which is basically electrostatic interaction between a negative and a positive charge. And within non-bonding charges, hydrogen bond is one of the strongest charge, and it helps maintain the entire structure of the complex itself. So if I can select just hydrogen and hide it, I'll also try showing you how, since we already viewed that DNA is such a highly negatively charged chain, that by default means that the thing that it's attracted to this protein complex should be a positively charged one as well. And hence I will just select residues. First of all, clear all the residues and select protein residues, and then we'll apply a electrostatic map to it. And here, as you can see, in the center of the protein complex there are some variations within each of the chain, and this variation actually helps the chain bind together itself, both the structural complementarity and the electrostatic interactions between them. As you can see, this chain has a negative region right here which is attracted to the positive region, and it forms this entire giant structure. But most importantly, the interactions between the protein and the DNA itself are highly electrostatic in nature, in which the negative charges of the DNA perfectly bind together to the positive charge of this protein. So if I hide the surface right here and have a look at the residues, you can see there's a nitrogen right here and again another nitrogen right here. Both of these are arginine and lysine, which majorly contributes to the positive charge nature of the protein itself at the boundary of its interaction with the DNA. And hence this nucleosome maintains a structure when the DNA bases lay dormant and is not utilized either for transcription or translation. And when the time comes when the DNA bases are actually required for translation, a process called acetylation and methylation by acetyl transferase and methyl transferase happens within the histone atoms. So as we all know, acetyl group is a group with basically a methyl group and a carboxy group. So it's CH3C double bond O. Uh, that oxygen actually confers it a very highly negative charge to the group. So if we add that acetyl group to any of the amino acid residues, usually it happens with hydroxyl group amino acids such as serine, threonine, and tyrosine. So that hydroxyl group reacts with the acetyl group and releases a water to form a covalent bond. So that confers a negative charge to the histone proteins. And hence, since negative and negative repels, the DNA slowly unwraps around the histone protein, and now the DNA is actually available for transcription using a RNA polymerase. And we can also try to view the hydrophobicity nature of the entire protein as well. And majorly, as you can see, since I told the interactions were electrostatic in nature, there is not much pattern that we can observe within the protein itself um, at the regions of the interaction. And similarly, DNA is a very hydrophilic entity, and the London forces, even though much weaker than the electrostatic and hydrogen bonding, does play a role between the hydrophilic uh, interaction of the DNA along with the hydrophobic regions of the protein as well. So first of all, let me hide the sphere and hide the atoms. And now focusing on the hydrogen bonding actually happening within the complex itself, which again is a complementarity to the electrostatic nature of the interaction between the DNA and the protein. First of all, I will deselect all of the entities and click on hydrogen bonds so that here we can view all the hydrogen bonds which are happening within the protein. And let me first hide all the hydrogen bonds and only select the nucleic acids and then turn on hydrogen bonding. Here we can clearly see the hydrogen bonding that's occurring within the protein chain, which is green in color here, along with the phosphate backbone of the DNA. So this negatively charged phosphate backbone actually bonds to a hydrogen bonds to the backbone of the residue, in which there's an electronegative atom, and hence the hydrogen possesses partial positive charge. And this nucleosome is a major factor in the compaction factor of the DNA in our cell. And a fun fact is that if we stretch out the all of the DNA present in one cell, it will stretch up to 2 meters in length. And hence all of those DNA should be packed within the volume of a cell, which is nearly a few nanometers or a few micrometers in diameter actually. And within the nucleus, the distance even reduces more. So this kind of compaction around the protein and the repeating compaction of the nucleosomes themselves, stack by stack, present as stacks, form the chromatin, which tightly binds nearly 3 billion nucleotide bases of our human DNA within such a small volume. And this genetic code helps us maintain our form and function, and is what makes us human essentially. Thank you.